Welcome to the revision course on Animal Nutrition Unit 2 brought to you by Dr. Pramod Kumar from GNP Sir Classes. First, let us talk about feeding experiments. For designing a feeding experiment, the experimental diets should be similar, the animals should be of similar age, size and sex, and a randomized pattern should be followed. The experiment should be sufficiently replicated. The different feeding experiments are comparative feeding trials. 2 or more rations are compared. Feeding trials with laboratory animals, done for pilot experiments, purified diet method, purified source of various nutrients, for laboratory animals, germ-free technique, free of contamination by bacteria, viruses, mold, protozoa, group feeding versus individual feeding, controlled feeding versus ad libitum feeding, equalized paired feeding or paired feeding, and slaughter experiments. Now we discuss the measurement of digestibility. So first discuss what is digestibility. Digestibility is the portion of feed or nutrient present in the feed that is not excreted in the feces of animals. It is expressed as a digestible nutrient or digestibility coefficient. The estimated digestibility is called apparent digestibility. Digestibility of feed is measured by in vivo, semi-in vivo, and in vitro methods. The formula of calculating digestibility of feed is shown here. In the in vivo method, the digestibility is calculated by the above formula. The semi-in vivo methods, the in sacco technique or Viver, in vivo artificial rumen, methods are used. Remember the bag size, paw size and sample size. In vitro methods utilize either the rumen liquor or enzymes or the Rusitech method. Now let's discuss the feeding standards. Feeding standards are the tables showing the quantities of nutrients to be fed to the various classes of livestock for different physiological functions like growth, maintenance, lactation, egg production and wool growth. The objectives of the feeding standards are to guide farmers to selecting properly balanced rations for their livestock. To classify different physiological functions like growth, maintenance, lactation, egg production and wool growth. Considering individual animal for preparation of FS due to their ability to digest and utilize feed. The table shows the classification of the feeding standards for livestock. First we discuss the comparative type feeding standards. 1. Hay standard. In 1810 German scientist Albert Thayer gave a concept of hay equivalent as measures of relative value based on determining the materials in feed extractable with water or other solvents. 2. Scandinavian Feed Unit Standard. In 1884, Professor Fjord formulated the Scandinavian feeding standard. In this system only one factor, namely, the feed unit was taken into account. The value of 1 pound of common grains such as corn, barley or wheat, is given as 1 unit value and the value of all other foods is based upon this. According to the standard, 1 feed unit is required for each 150 pounds of body weight and an additional feed unit for every 3 pounds of milk production. Feeding Standard 2, Digestible Nutrient Type. 1. Gruen Feeding Standard. In 1859 Gruen, a German chemist, published first feeding standard for farm animals with crude protein, carbohydrates and fat contained in the feed as the basis of the standard. 2. Wolf Feeding Standard. In 1864 Dr. Emil von Wolf proposed a standard based on digestible protein, digestible carbohydrates and digestible fats contained in a feeding stuff. He calculated the ratio of digestible protein to digestible carbohydrate, called the albuminoid ratio, because he recognized that the proportion of protein in the ration affected its digestibility. Later on, albuminoid ratio was developed as a nutritive ratio. The nutritive ratio is the sum of the digestible carbohydrate, digestible protein, and 2.25 times digestible fat, divided by digestible protein. 3. Atwater S Available Fuel Value. In 1890 Atwater proposed a feeding standard based on the available fuel values of the feeds. The system is based on the heats of combustion of protein, fat and carbohydrate, which are corrected for losses in digestion, absorption and urinary excretion of urea. 4. Haker's feeding standard first time considered quantity as well as the quality of milk produced in formulating a feeding standard. His standard included digestible crude protein, carbohydrates and fats. Later it was expressed in digestible crude protein and total digestible nutrients. 5. Savage feeding standard. He expressed his standard in terms of DCP and TDN and further showed that about 2/3 requirement of the dry matter should be met by feeding roughages and the remaining 1/3 from concentrates. 6. Morrison feeding standard. His standard were first presented in the 15th edition of Feeds and Feedings, published in 1915 and were then called Modified Wolf and Lehman Standard. They soon came to be known as the Morrison feeding standard. These standards have expressed in terms of dry matter, DM, digestible protein, DP, and total digestible nutrients, TDN. Morrison indicated the nutrient requirement of animals in a range rather than in one figure. The average of Morrison standards has been accepted for Indian livestock. 7. National Research Council, NRC, standard. In 1942, the Committee on Animal Nutrition of the National Research Council, NRC, of the National Academy of Sciences published the recommended nutrient allowances for farm animals, comprising separate reports for poultry, swine, dairy cattle, beef cattle, sheep, and horses. The standard includes digestible protein and total digestible nutrients and also includes the recommended requirements for calcium, phosphorus, carotene and vitamin D for dairy cattle, beef cattle, pigs, poultry, sheepdogs, horses, laboratory animals etc. 8. Indian standards. Dr. K.C. Sen, the first director, National Dairy Research Institute, Bangalore and Karnal and Ray have compiled the feeding standards for zebu cattle and buffaloes, based on Morrison's recommendations, where they adopted the average of maximum and minimum values recommended by Morrison. 9. Indian Council of Agricultural Research Feeding Standard. These standards were updated and were revised in 1998. Latest feeding standard was published by ICAR, New Delhi in 2013. In India, Bureau of India Standards, BIS, also publishes feeding standard for different categories of poultry. Latest BIS publication for poultry feeding is 2007. The feeding standards are based on the experimental results and have been organized to contain information on daily DM, DCP, TDN, calcium and phosphorus intake in cattle. For poultry, protein is expressed in terms of crude protein, %, whereas energy is expressed in metabolizable energy, kilocalorie per kilogram diet. Feeding standard, production value type, 1. Kellner feeding standard. In 1907, Kellner, a German scientist, investigated a feeding standard based upon starch as the unit of measurement. He took into account not only the digestibility of the feeds as calculated from the amount lost in feces and urine but also the entire loss from the body including energy expended in digestion and passing the food inside the body, chewing, etc. 2. Armsby feeding standard. Armsby expresses his standard in two factors, that is true protein and terms of net energy. Armsby determined the net energy required for mastication, digestion, assimilation and also the amount of heat and gases given off through the excretory channels. Agricultural Research Council, ARC, standard. In United Kingdom, a technical committee was set up to develop the standards in 1959 by the Agricultural Research Council, ARC, which later came to be known as Agricultural and Food Research Council, AFRC. For ruminants, protein requirements are expressed in terms of metabolizable protein, G, and energy in terms of metabolizable energy, MJ. Limitation of feeding standards. Feeding standard is not a complete guide to feeding because other factors such as palatability and the physical nature of the ration are not taken into account. Now we discuss the minerals. Minerals are the inorganic elements that are essential for the animal's physiological functions and metabolic processes. Their presence is essential for maintaining life and animal health as they contribute to proper growth, overall health and disease prevention. The mineral matter constitutes about 4% of the animal body's weight. All animal tissues and all feeds contain inorganic or mineral elements in widely varying amounts and proportions. This table shows the various minerals. Let's discuss the functions of minerals. Structural functions. Calcium, phosphorus and magnesium are essential components of the skeleton and sulfur is necessary for the synthesis of structural proteins. Catalytic functions. Some mineral elements are firmly bound to the proteins of enzymes, while others are present in prosthetic groups in chelate form. They can act as catalysts in enzyme and hormone systems as components of metalloenzymes, hormones. Regulatory functions. Certain elements have a regulatory function in controlling cell replication and differentiation. Zinc acts in this way by influencing the transcription process. Physiological functions. Sodium, potassium and chlorine have primarily an electrochemical or physiological function and are concerned with the maintenance of acid-base balance, membrane permeability and the osmotic control of water distribution within the body. Now we talk about the classification of minerals. Minerals that are needed in relatively large amounts are referred to as major or macro minerals. Others that are needed in very small amounts are referred to as trace or micro minerals. The major minerals are required in concentrations of greater than 100 ppm, parts per million, and often as a percentage of the diet, or gram per kilogram, while trace elements are required at less than 100 ppm. This table shows the macro and micro minerals, newer microelements, and the toxic minerals. So first we discuss calcium and phosphorus. About 4% of the animal body consist of minerals. Over 70% of the ash of the body consist of calcium and phosphorus. Approximately 99% of the calcium and 80% of the phosphorus of the body are present in the bones and teeth as a compound called hydroxyapatite. The other 1% of Ca is distributed in cellular fluids, where they are involved in different metabolic and physiologic activities. Calcium, Ca, and phosphorus, P, are considered together because they are closely associated with each other in metabolism. A deficiency or an excess of one will interfere with the proper utilization of the other. Calcium, Ca, and phosphorus, P, are the two most abundant mineral elements in the animal body. In bones, the ratio of Ca, P is 2:1. The absorption of Ca and P occurs from most of the intestinal tract, with the duodenum and jejunum being the most active absorptive sites. Animals absorb Ca from their gut according to need, and they can alter the efficiency of absorption to meet the change in requirement. Plasma calcium varies from 9 to 12 mg per deciliter. Plasma phosphorus varies from 4 to 9 mg per deciliter. Now let's discuss calcium in some more detail. Calcium is the most abundant mineral element in the animal body. It is an important constituent of the skeleton and teeth. It helps in muscle contraction, myocardial and neuromuscular excitability, activation of various enzymes, blood coagulation and secretion of several hormones. Deficiency symptoms. In young animals, rickets, rachitic rosary, poor bone development, effortlessly bending and misshapen bones. In adults, osteomalacia, general softening of the bone. Osteoporosis, bone reabsorption is more than bone formation. Milk fever, parturiente paresis, catetany, in high yielders. Thin and fragile eggshell in birds. Nutritional secondary hypoparathyroidism, NSH. Osteodystrophy fibrosa and big head disease in horse. Let me explain the regulation of blood calcium levels. Ca is regulated by the action of parathyroid hormone, PTH, calcitonin and active forms of vitamin D. When blood Ca is low, hypocalcemia, PTH is released from the parathyroid gland, which leads to increased Ca and P resorption from bone, increased P excretion into urine and an increase in absorption of dietary Ca from the gastrointestinal tract. When blood Ca is high, hypercalcemia, another hormone called calcitonin is released by the parafollicular cells of the thyroid gland. Calcitonin reverses PTH functions to lower blood Ca level to normal by decreasing calcium mobilization from bones. PTH also plays an important role in regulating the amount of Ca absorbed from the intestine by influencing the production of 1,25-dihydroxycholecalciferol, a derivative of vitamin D, which is concerned with the formation of calcium-binding protein. Finally, the hormone stimulates the kidney to resorb urinary calcium. This chart shows calcium homeostasis and hope it helps in understanding the calcium homeostasis. This chart shows the stimulus, the hormone responsible, and the result. Now we discuss another important micromineral, phosphorus. Phosphorus is a part of phosphoproteins, nucleic acids and phospholipids. It plays a vital role in energy metabolism in the formation of sugar phosphates, DNA and RNA, and adenosine D and triphosphates, ADP and ATP. Also, it is part of cell membrane phospholipids that are involved in maintaining cellular fluidity and transport of nutrients into cells. Cereal grains are rich in phosphorus. However, P in cereal grains are present in the bound form as phytate or phytic acid. The availability of P from such bound sources varies from 20% to 60%. Monogastric animals lack the enzyme phytase to release them from the bound form, and the term available P is commonly used to designate unbound forms of P in the diet of monogastric animals. Ruminant animals produce microbial phytase enzyme that can split and liberate phosphorus. Phosphorus deficiency symptoms: pica, depraved appetite, eating of bones, wood, clothing, soil, plastic, etc. by animal. Reduced fertility and delayed conception. The control of P metabolism is different from that of Ca. If it is in an available form, P is absorbed well even when there is an excess over the requirement. The excess is excreted via the kidney or the gut, via saliva. In monogastric animals, the kidney is the primary route of excretion. Plasma P diffuses into saliva, and in ruminants the large amount of chewing during rumination results in saliva being the major input of P into the rumen rather than the food. This chart shows the phosphorus metabolism in the dairy cow. The combined effects of calcium and phosphorus are shown in this table. Another closely related micromineral to calcium and phosphorus is magnesium. Magnesium is closely associated with Ca and P. About 70% of the total magnesium, Mg, is found in the skeleton, but the remainder, which is distributed in the soft tissues and fluids, is of crucial importance to the well-being of the animal. It is essential for bones and teeth, activator of several enzymes transferring phosphate from ADP to ATP, cofactor for decarboxylation peptidases, alkaline and acid phosphatases. Deficiency symptoms: Ca deposition in the kidney, renal calculi, panting and gasping breathing in chicks, grass tetany, grass staggers, in calves, fed for long periods on milk alone called as milk tetany, and in cows, fed on lush green pasture called as wheat pasture tetany, and lactation tetany, usually accompanied with hypocalcemia. Mg is absorbed from the small intestine in pre-ruminant and monogastric animals, and largely from the rumen in ruminants. Active absorption occurs via a sodium-dependent transporter, and there is also some passive diffusion. Active absorption from the rumen is impaired by high potassium and low sodium in the diet. For ease of remembrance, this table depicts the functions, deficiency disease, and toxicity symptoms of magnesium. So that's all for today's lecture. We will discuss the micronutrients in our next class. Thanks for watching this video. Do not forget to share, comment, like and subscribe GNP Sir official YouTube channel. Bye.